All right, y'all. Hope you're having a great day. Today we're going to cover the reproductive system. There's a chance this recording might come in two parts. So I'm a little crunched on time, and if I don't have enough time to finish this whole chapter right now, then I might cut it after the male repro and then have female repro in a different recording. So if you don't have everything for this recording, then definitely look for the second one that would probably be labeled female repro, and this would be labeled male repro. If this is the only recording, then it would probably be just labeled the repro system. So, all right, let's start talking about the reproductive system. Obviously, what we're talking about are the this jumped several slides here. So let's come to here. So first off, this reproductive systems, the real key is this is going to provide a means of producing the next generation and eventually continuing the species. And so that's really what the reproductive system's primary function is to continue our species overall by producing that next generation. So parts of reproductive system. First off, I want you to realize that there are four components overall of the reproductive system. Let's kind of talk about their functions a little bit. First off, especially whenever we think about lab, kind of relate this to lab, and it will make a little bit more sense as well. Gonads. So the gonads are the sex organs. This is where the sperm and the egg are created, and the proper term for the sperm is spermatozoa. The proper term for the egg is the secondary oocyte. And so the gonads are where the gametes are going to be created. And don't forget the word gamete is just the term for a sex cell for either that spermatozoa or that oocyte. And so not only is it where these gametes are made, but it's also where the sex hormones tend to be made, the primary sex hormones. So this is where testosterone is made. This is where estrogen and progesterone are going to be made. And so they're in the gonads. They've got two functions really: to produce the gametes, but also to produce the sex hormones. Now we're going to talk here in a second about gametes and how they have half of the genetic information, half of the DNA. So these are haploid cells. So we'll talk about that. And that way, if each one has a half, when they unite to create that zygote, the first cell after fertilization, then now that first cell now has a new complete set of DNA that's never been found before on our planet, right? So that's kind of cool. The reproductive tract is the next structure. The reproductive tract tends to transport the gamete, the gametes from where they're created to where they're to unite. And so the reproductive tract in the male is beginning with the epididymis and the vas deferens, and then out through the ejaculatory duct into the urethra. And then for the female, the reproductive tract, it begins with the uterine tube into the uterus and then out the cervix and into the vagina. And so those areas are the tracts that lead to and from basically the gonads. And this is the passageway that these gametes are going to travel in order to fertilize. The accessory structures. So these are especially more prevalent in males than females. So accessory structures, especially in males, provided extra fluid, and each one of the fluids provides a unique function for the semen overall. And so accessory structures are going to add extra fluids. They're going to add some new feature to the reproductive system. So females also have other accessory structures that we don't talk about, certain glands that release secretions that we just don't talk about in this presentation today, because I'm kind of cutting things short, especially for this abridged semester. The final component are the external genitalia. This is anything basically involved with reproduction that is visible on the outside of the body. So for the male, obviously we're talking about the penis, and then the scrotum contains the testes, but mainly we're referring to the penis. And whenever we talk about the female, then the term is vulva to describe all of it combined. But remember, that's not one point. That's not a specific part on the female anatomy. That the vulva includes components like the labia majora, the labia minora, and the clitoris. So those are some of its major components that are on the outside of the female body. Now, let's talk about this reproductive cycle, and let's talk about the processes of mitosis. First, let's don't forget, mitosis is a cell division that creates exact copies of cells. So mitosis is usually used for growth and repair, but here we're going to see it used by the stem cells in the reproductive system to create the cell that will then go through meiosis and create the gametes or the sex cells. So mitosis makes exact copies. Meiosis, some people call it meiosis or meiosis. This is a cell division that is going to produce cells with half of the DNA. A normal cell that has a full set of chromosomes and DNA is called a diploid cell. Diploid. Diploid. That's our normal body cells. But the gametes are unique. The sex cells, the spermatozoa and the secondary oocyte, only have half of the DNA compared to that diploid cell. So we call these cells haploid. Again, I talked about it just a second ago. This is so that when the sperm and the egg unite, then each contributes half of the DNA and they create a brand new whole. And so we have to create these sex cells, and these gametes are produced through that process of meiosis. Now, meiosis takes place in the gonads. So an adult has gonads. They're going to produce the sex cells. They're going to produce gametes. Don't forget that's the term for the sex cells at fertilization. So, with the actions of reproduction, then comes fertilization. And so, at fertilization, the sperm unites with the egg. And now we create the first diploid cell of that brand new organism. And I mentioned that just a second ago. That's called a zygote. Zygote. So that zygote, then it is going to undergo a lot of rapid mitosis. It's going to grow. And so the zygote is going to change quickly and it's going to grow many, many cells. I want you to understand that the zygote is eventually going to become an embryo. So an embryo is what we refer to in the first 12 or so weeks of development of just about any creature. But the beginning stages, an embryo means that we all look the same in this stage of development. Once we start to look like the adult, then that's when we're called a fetus. So for us, it's the first trimester that is embryonic, and then the last two trimesters are only when, in those areas, in those times, is when the body starts to look more like an actual human. And so that's when we refer to it as the fetal stages. So again, we're going to see this mitosis, we're going to see this growth. This baby is going to be born and it's going to grow through mitosis, and eventually that baby is going to have gonads. And in the gonads, that adult eventually is going to produce gametes, and their cycle is just going to continue. So we're seeing this cycle of meiosis creating haploid cells, and then fertilization fusing two haploid cells and creating a new diploid cell that makes the new organism before that one grows up and repeats the process, repeats this cycle. Again, we're going to focus on male first, and then we'll look at female. Male reproductive is the process of creating sperm. All processes combined is spermatogenesis. Now, just looking on this page, I see what I like to refer to as a lot of sperm terms. So here we see the term spermatogenesis. Here we see the term spermiogenesis. Here we see the term spermiation. I've already mentioned spermatozoa, and we're going to talk about spermatocytes and spermatogonia. So there's a lot of sperm terms. So you need to make sure that you give yourself a list and you completely understand the definitions of these. So when we use these words, they make sense to you. Right? They make sense and you understand what's actually happening. Again, spermatogenesis is the overall process of sperm production. There are three individual processes, or three stages in this. First is mitosis. I want you to understand that mitosis is what happens with the stem cell. The stem cell is called a spermatogonia or spermatogonium. Either an IA or a UM at the end is fine. So spermatogonia, G-O-N, spermatogonia or spermatogonium is going to divide, and what it's going to create is called the primary spermatocyte. So the primary spermatocyte enters meiosis. So the primary spermatocyte enters meiosis, and we tend to kind of correlate primary spermatocyte with meiosis 1, and then secondary spermatocyte with meiosis 2. And eventually, the secondary spermatocyte is going to become an immature sperm called a spermatid. So when that spermatid, and the spermatid's going to, it's still kind of inside of a cell, but it's finally starting to grow its flagellum, finally starting to grow its tail. So it's kind of got a little nub. And so that's how we can tell the difference when we look at the cells. We look inside and we kind of start to see a little nub forming either on the inside or the outside. And that means that that's a spermatid, but it's not ready. It's not a real tadpole. It's not a real spermatozoa yet. So meiosis takes that primary spermatocyte and divides it and creates the spermatid. The spermatid then enters the process of spermiogenesis. Spermiogenesis is where the spermatid finishes its maturation, it finishes growing up, and it becomes a spermatozoa. Now, it's going to become a spermatozoa, and that is the actual sperm that's ejaculated out of the male. But this spermatozoa is not activated yet. So at the end of spermiogenesis, we have these sperm cells called spermatozoa, the proper term for the sperm. And these are in kind of a suspended state of animation. They're not doing anything yet. They're waiting to get some sugar to be activated. They need some sort of carbohydrate, some energy source to be activated. And so not until they reach that do they actually become activated, because they only have a short time, a few days to live. Now, inside all of this is taking place inside of what's called a seminiferous tubule. Here is inside of a seminiferous tubule. The spermatogonia are towards the outside, and then we have these primary and secondary spermatocytes. We see them listed as dividing. Then we see the spermatids, small mini versions of true spermatozoa, which are these actual tadpole-looking structures. Now, when we look at these spermatozoa right here, we notice that they're stuck in a purple goo. There's a term on that previous slide called spermiation, and I'll go back to it. Spermiation is where these spermatozoa wiggle free. They are released from this goo, enter the inside of the tube, which means they're on their way out of the testes. And so the process of spermiation is when the spermatozoa are released from that fluid. The spermiation is the process where the spermatozoa are released from that fluid, and they can start to flow out through the lumen of the seminiferous tubule and start to flow out of the testes. So this is kind of the last process. After spermiogenesis, the last process is spermiation, and where they wiggle free of purple jelly that they've been living inside. Now, who made that purple jelly? Called Sertoli cells, or the sustentacular cells. So either Sertoli or sustentacular cells is the term that we're going to use. So the sustentacular cells, I'm sorry guys, I'm having some issues with my if it gets really bad, then I'll restart this. Hopefully that's going to be the last of it. So these Sertoli, or these sustentacular cells, these guys are kind of like nurse cells. These are the guys that are providing that purple jelly that's surrounding the developing spermatozoa, and it's giving it its nutrients, and it's kind of protecting it. And so here we can see, let me zoom in even more. Here we can see the sustentacular or Sertoli cells. Here we can see the fluid that they're making and how it's surrounding all these cells, cutting them off from the bloodstream, but also kind of dictating what's going to happen by providing the nutrients and taking away the waste. And here we see those final spermatozoa that have become the actual sperm, and they're going to wiggle free through spermiation, and that's going to release them into the inside of the tube to be released out. Now again, this what we're looking at is a typical meiosis, typical meiotic division. Males are constantly doing this. It begins at puberty and it doesn't stop until death. Here again, mitosis begins with the stem cell making a copy, and that copy is going to enter the division. We're going to keep the original and use the copy. And so we put the copy through these divisions. First, meiosis is, again, the spermatogonium is going to make the primary spermatocyte. Primary spermatocyte then starts to become the secondary spermatocyte, and then it comes the spermatids. And the spermatids are going to undergo spermiogenesis in order to become the mature spermatozoa. Here we see what the typical meiosis produces. One cell starts and it makes 4 daughter cells. Even though these are males, we still call them daughter cells. And these 4 daughter cells are now haploid. They've got half the genetic information. And so if you notice right here, it doubled its DNA. And then it split it in half again, and then half again. And that's how we get 4 from 1, is because we actually take 1 and turn it into a double copy, and then we split that double twice to get half DNA in each of these. Okay, so this is your typical. Okay, this is what most meiosis is. Begins with 1, ends with 4, and they're all randomly different. All right, and they're haploid. Now, those tentacular Sertoli cells, as I've already mentioned, these guys help to promote the developing spermatocytes. So these guys are going to create that goo that surrounds them and kind of protects them and gives them their nourishment and takes away their waste. It also, that surrounding helps prevent them, and I mentioned this just a second ago, from being in contact with the bloodstream. Don't forget that these sperm are created in a suspended state of animation. They don't become active until they meet up with sugar later on in the reproductive tract. And so if they reach the bloodstream, they can access glucose, which is readily available, and then they would become active. And then when they're ejaculated out of the male's body, they would be dead, and that's a fertility problem. So those sustentacular cells, that goo that they embed around all those cells, that actually prevents them from in contact with the bloodstream and having that adverse reaction to that glucose, something that's just normal, but it would be bad if it happened then. Let's talk about the spermatozoa or spermatozoan. Okay, spermatozoa or zoan, singular versus plural. Let's talk about its anatomy. Here, notice, this is what you're going to take notes on, and I'm going to flip to a picture and show you. Head, part of the head is called the acrosomal cap. So you can give yourself that note. The head is the first thing, and I want you to know, and I'll point it out, we'll go ahead and do notes here. The head contains the DNA. Okay, so the head contains the DNA. The acrosomal cap is the cover of the head, and the acrosomal cap contains enzymes for penetrating the egg. The egg is coated in a thick mucus, and it has, a cell membrane. So in order to get into the inside of that cell, we need some enzymes that can eat its way through those layers. So the acrosomal cap is kind of the covering of the head, and it helps with that process of fertilization. The neck and the middle piece is where we have mitochondria. So this is where we're going to use energy to produce movement. And the movement that we're going to produce is with the tail, which is the flagellum, or flagella is plural, flagellum is singular. And so again, there's no energy reserves here. This structure is suspendedly animated until it gets those energy reserves, and that's when it becomes active. And it's a very fragile cell, and it only lasts a few days. And so it waits until the split second before ejaculation out of the male's body to become active. And so it waits until then to meet up with those sugars to actually start that process. Now here, let's take a look. Here is simply week 1 to week 5. So it takes about a little over a month to produce a spermatozoa. Here we can see the head. When we look at the head, the DNA is inside, and that's the whole key. What is this thing? What is a spermatozoa? It's nothing but DNA that can move, right? That's what we're seeing. It's mobile DNA. And so here we see this nucleus, and that contains the DNA, and surrounding the covering of the head is that acrosomal cap. And here we see that's where those enzymes are going to be located that can help penetrate that egg. The neck and the middle piece, we're going to have mitochondria that are going to help with movement. And the movement is from the flagellum, which is the singular version of a flagella, that tail. Now, let's look at the accessory structures, and then we'll kind of wrap this up for the male. First off, the epididymis is on the back side of the testes. As I mentioned in lab, it monitors the fluid and it adjusts the fluid that's leaving the testes. There's not very much fluid, maybe 5% of what reaches the final ejaculate, but really what it's doing is it's checking out the spermatozoa, the spermatozoans, right? It's looking damaged. It's looking to see, is there one that's not going to be able to fertilize an egg, and if so, it needs to be destroyed, needs to be recycled. If they are in good shape, then it's going to store and protect them, allow them to kind of finish their development a little bit for another couple weeks. And so maybe 2 weeks in the epididymis before they move on, and they line up and kind of wait in the vas deferens or the ductus deferens. So the vas deferens or the ductus deferens, that's their primary function, is to store the sperm, because sperm is not always being emitted. And so obviously it needs to be stored. And so the sperm is always being created, but it needs to be stored until it's ejaculated out of the male's body. And so it's stored inside of the vas deferens. So it's really just like a long line of sperm just waiting to be called on to be released. And if during this time that it's waiting for up to a couple months, if it starts to break down, if that sperm starts to become non-functioning, or it's been there too long, then it's just going to be absorbed by the vas deferens, and we're going to destroy it. We're going to try to recycle it and rebuild it later. Okay, so it stores, but it's also kind of an area where if the ejaculate is not removed, then or if the spermatozoa are not removed, then they're going to be recovered. They're going to be kind of destroyed and recycled, and they're going to allow fresh sperm that has a better potential for fertilization to move into their place. At the point of ejaculation, the vas deferens are going to help propel the sperm out of the male's body. Here you see the testes. The seminiferous tubules are where we're going to make the spermatozoa, and we just saw that kind of rounded circular tube that had development from the outside in, and we saw that kind of purple jelly surrounding those cells. So that's what's inside of here. That collects into the epididymis. The epididymis is just kind of a coiled up mass of tubes on the back side of the testis. Here is where it's going to do quality control. It's going to act like a nursery, allow it to kind of finish its develop, store, and protect, and then it's going to lead into the vas deferens. So here is the vas deferens, and the vas deferens is eventually going to puncture into the prostate at the ejaculatory duct. And here we can see the end of the vas deferens. The end of the vas deferens meets up with the seminal vesicle, and that's going to be our next accessory structure. And there they combine their fluids. And at that point, they're going to leave through the ejaculatory duct combined before entering into the urethra. Right? Now they go through the ejaculatory duct and through the prostate. So there is the prostate gland, and then below it we see the bulbourethral gland. And so this is one that's going to produce a thicker mucus. And here we can see the root and the body of the penis. And these are things that we talked about in lab. Now, the seminal vesicle or the seminal gland does produce the majority of the fluid in semen, and that's the reason that we call semen. Semen is because it comes from the seminal vesicle. So 60%, so that's a majority. Sugar, fructose, so this is going to activate the sperm. This is going to make the sperm mobile, and this is not going to happen until emission, until the split second before ejaculation. So that fructose is important. Some of these prostaglandins are going to help stimulate some smooth muscle contractions in the male to help propel the sperm out, but also once that enters the female, trigger her smooth muscle to move that sperm closer to her egg. And so this is going to increase again the chances for fertilization. Not only do these smooth muscles contract to help kind of move things along, but also we're going to include fibrinogens, and these are good for clotting. And so the semen, when it is ejaculated out of the male, it should clot. It should clot very quickly, and then it should reliquefy later. Okay, so it should take about 30 minutes to reliquefy, and that's actually a fertility test. And if that doesn't happen, then that can be the cause of infertility. So this is going to cause the semen to clot inside the female, and what that does is it increases the chances of the sperm uniting with the egg. It's going to prevent the sperm from swimming out of the female, right? From exiting the female. So it's going to increase the time of contact to increase the overall chances of fertilization. Again, both of these kind of ejaculatory duct, they're going to meet up. So at that split second of ejaculation right before, then these are going to meet up, activate the sperm. Then we're going to gain some fluids from the prostate gland. So let's talk about that. So the prostate contributes about a quarter of the total fluid, and its primary function is it has a secretion that helps prevent urinary tract infections. So UTIs obviously are a little bit more prevalent, or a lot more prevalent in females, and this is one of the main reasons is that females do not have a prostate gland, and so as a result, they don't have something that's kind of combating that problem of bacterial infection. Now, the CALPers, or the bulbourethral glands, the CALPers or bulbourethral glands, these guys are located in that urogenital diaphragm. They're kind of located in that membrane of muscle, and that's kind of surrounding that membranous urethra. And so these guys are going to produce a very thick mucus, and part of the function of this mucus is it is going to help to neutralize some of the acids that are in the urethra. And so the residue from urine that has passed through that urethra, because now we're going to share that passageway with the repro system, we don't want those acids to kill these sperm while they're on their way out. And so as a result, this is going to usually come through, this fluid tends to come through before the sperm come through. And so before the rest of the semen and the spermatozoa are going to come through the urethra, this is going to come through, it's going to kind of clean it up to make sure that it can't damage those spermatozoa, and it's going to lubricate the urethra so that the sperm can get out, and it will also lubricate the glands of the penis, and this is mainly for the initial actions of penetration. So many times this fluid comes out first, and sometimes this is referred to as the pre-ejaculate. Now, when we look at semen and try to do a semen analysis, we're looking for some basic things inside of semen. Obviously, there should be a spermatozoa, right? So there should be spermatozoa. There should be seminal fluid, right? So we know these things are two required for this fluid, but also there should be some enzymes involved. So many times we're checking to see if all three of those are in that fluid, that ejaculate. Now, for a fertility test, let's talk about a semen analysis. For a fertility test, the semen need to pass 5 checks. The first one is volume. Okay, so how much, right? So that's one thing that needs to be considered, is how much. 2 to 5 milliliters is the average ejaculate. So less than that could indicate a fertility problem. Sperm. So how many sperm are in there? The spermatozoa count, right? So how many sperm are in each milliliter? So here we're going to try to do per milliliter, and then that way we can make sure that we keep it relative. So we can see, is there a lot or is there a little in that same space that we're always measuring, okay? Something else that we're going to look for is shape, okay? The shape of the sperm, make sure that they are shaped right, and also make sure that they are mobile, okay? Make sure that they can move. And these are two of the primary things that are going to be monitored. If more than 40% have problems with their shape or their mobility, then that is going to lead to a fertility problem. Okay. One of the other things that we're going to see in a fertility test is going to be what's called liquefaction. Liquefaction is that ability for that sperm excuse me, for that semen to coagulate and then liquefy again later. So if that takes place at the usual 30-minute or so timing, then everything tends to be okay. Now, if all of those check out and there's no issues with any of this, then there is one last test that I like to mention that might be the issue. And it might be used in order to figure out if it's the male's fault. Now, obviously, it's always easiest to check a male than it is to check a female. Whenever we think about this process, to check a female is very invasive to get inside of ovary and to harvest one of her eggs, right? But to get a sample from a male, we simply need to get a sample of the ejaculate. So again, that's a magazine, 5-10 minutes in another room. No, just kidding. But in all seriousness, it's a lot easier to get a sample from a male. So we're going to test the heck out of that male sample first before we have to do any of that super invasive testing for the female. So the last test, if all these things checked out, then it's called the hamster test. Okay, and I don't want your brains to wander, but it's called the hamster test. The hamster test: a human sperm will fertilize a hamster egg. That's freaky, right? A human sperm will fertilize a hamster egg. We can look at this under a microscope and observe fertilization. The key: the egg will not develop. It will not turn into a little hamster human. Okay, so don't worry about little rat people running around. That's not going to happen. But the key is, we've learned that in a lab these will fertilize each other, or the human sperm will fertilize a hamster egg. So if we visibly in a petri dish take the male's sperm and with that hamster egg and we see fertilization, then we know that his sperm and his levels are checking out, then he is capable of fertilization. So then it's time to turn the efforts to the female and start looking at maybe what they can do to help her in order to increase that fertility. So again, here are those accessory structures that we just mentioned. Not really going to spend too much time. Here is the penis, and there is that urethra. So this is a good view of the urethra. Here is the penis, the body, the glands, and then the root is back behind the inside of the body of the male. The last thing that we're going to talk about before I cut this presentation off, and I will be cutting this into two because this is taking me a little bit longer than I thought because I had some issues there at the beginning. So this is just going to be for male repro, and then I will post a shorter female repro recording as well here before too long. First off, male sex hormones. Let's talk about the sex hormones. GnRH, Guns N' Roses hormone. No, I'm just kidding. GnRH is gonadotrophic releasing hormone. Just know it as GnRH. Gonadotrophic releasing hormone stimulates the release of these two hormones. And these are two of the primary hormones in both males and females that do different things. And then we have the main sex hormone. So these first two, FSH and LH, follicle stimulating hormone, in males is going to trigger sperm production. And so follicle stimulating hormone in females stimulates her follicles to develop her eggs. So this is really named after its female function, but knowing that it stimulates the egg production, it kind of does the same in males. Instead of the egg, it stimulates sperm production. Okay, so it triggers spermatogenesis. And so does testosterone, which is our main androgen. Now, LH is luteinizing hormone, L-E-U-T. Luteinizing hormone is in females, this is what causes ovulation, but here in males, LH triggers the release of testosterone. Again, testosterone is the primary androgen. This is the main male sex hormone, and its primary function is to trigger sperm production, right? That's really what it's for, but it's also going to have some other effects. It talks about secondary sex characteristics, but it's going to have several effects throughout the body, and all these can kind of be considered secondary sex characteristics. It affects the central nervous system. It increases the sex drive. More testosterone makes men think about sex and have a higher sex drive than females overall, right? So this is part of the theory that testosterone levels are important for. This is another important function. Testosterone stimulates muscle mass. It stimulates muscle metabolism. So men tend to have bulkier, bigger muscles than females do. If you ever look at bodybuilders, you can see some guys could get super huge, but the females, they couldn't get that big ever, even if they tried really, you know? And so they would have to take testosterone to get that big. And so stimulating muscle metabolism, males have more muscle mass as a result. And so there's also secondary sex characteristics like a lower pitched voice, hair on the face, broader shoulders, taller overall height due to bone growth. So there's various things that are considered to be groups of characteristics dealing with secondary sex characteristics. Now, as I mentioned, I don't have enough time to finish this up right now because I'm in a little time frame where I have to start another class here in a second. But I wanted to go ahead and record this half of the lecture. And now I'm going to stop this recording. And whenever we come back, the next recording is going to pick up right here with physiology of the female reproductive organs. And we're going to talk about oogenesis and the process of producing eggs. So go ahead and turn this one off and be on the lookout for the female reproductive recording. Thanks, y'all. Have a great afternoon.